Hello everyone, hope you're all doing good. This video is looking at polysaccharides, and the three that we're going to focus on are starch, glycogen and cellulose. For all exam boards, you pretty much have to know their structure and relate it back to their function. That is basically how is it that their structure allows them to carry out the role that they play in organisms. By definition, polysaccharides are complex carbohydrates. They're straight or branched chains of many monosaccharides. All of these three contain only glucose as the monosaccharides, but their properties are very different. The differences are a function of the glucose isomer involved, so whether it's alpha or whether it's beta, and the types of linkages joining them. We're going to look at starch first. Starch is an energy storage molecule found in plants, and it's found in concentrated amounts in insoluble starch granules within specialized plastids called amyloplasts in the polymer of alpha glucose and consists of two types of molecules, amylose and amylopectin. The key point here is about the insolubility of starch. As it is such a large molecule, it is known as being insoluble. This is a good feature to have as it does not affect water potential in the cells. This basically means that water cannot enter the cell by osmosis in excess and cause the cell to burst, which makes starch a really good energy storage molecule. If you're making notes as we go along, I'd highly recommend that you pause the video right now and write this point down. So I think we need to talk a little bit about amylose and amylopectin. We'll start with amylose first. There are a few key points that we need to understand. Firstly, amylose makes up about 25 to 30% of the starch molecule. The rest is amylopectin. Amylose is a long, unbranched chain of alpha glucose molecules. Remember from my video on formation of glycosidic bonds when we talked about condensation reactions? Well, amylose will form when you have many alpha glucose molecules all forming glycosidic bonds between themselves to form a chain like you can see on that diagram. We call these bonds 1,4, meaning that the bond occurs between the carbon 1 of one molecule and the fourth carbon of another. This leads to amylose having a coiled structure, like you can see in that diagram. It's almost like a spring, and so we can say it's compact. And when molecules are compact, it means that they're good for storage because they can fit in small spaces. If you struggle to understand this concept, think about a ball of wool. When we buy this wool, it's tightly wound up around itself to make it easier to store at home or to use when knitting. If you wanted to store this ball of wool, it's much easier to do it in its smaller compact shape as it takes up less space. Amylose is also a bit like this slinky in terms of its shape. Lots of alpha glucose in their 1,4 bonds cause a twisting in the shape and creates this coiled shape, much like the one that we see floating around on the screen. In the exam, you can refer to this shape as a coiled helix. Next is amylopectin. Amylopectin makes up about 70 to 75% of a starch molecule. It's a long and branched chain of alpha glucoses. The long chains arise with the usual 1,4 bonds that we saw in amylose. The branches arrive from 1,6 bonds. This means there are glycosidic bonds between the carbon 1 of a glucose molecule and the carbon 6 of another glucose molecule. The diagram here illustrates the bonds a bit better, but I'll zoom into it in a second to show you this. These branches that are created are sometimes known as side branches, and they allow enzymes to break down the molecules as they can get to the bonds easily. The branches also allows many millions of glucose molecules to be stored in a compact form because each branch will coil up like amylose does to get into its slinky type shape or its coiled helix. This diagram here is just a zoomed in version showing the 1,4 and the 1,6 bonds in closer detail, as well as the locations of those glycosidic bonds. The next polysaccharide that you need to know about is cellulose. Cellulose is the structural material found in the cell walls of plants. Cellulose is an unbranched polymer of beta glucose molecules bonded by 1,4 bonds. As many as 10,000 glucose molecules may be linked together to form a straight chain, and they then form parallel chains of cellulose. They become cross-linked with hydrogen bonds and form bundles of around 60 to 70 molecules, and these bundles are known as microfibrils. These microfibrils are very, very strong and, of course, are a major component of plants in cell walls. Only a few organisms can digest cellulose. It's an ideal structural material. This diagram shows the formation of those individual chains of cellulose and then a bundle of microfibrils and eventually the cell wall of plants. I always use an analogy of uncooked spaghetti to illustrate how cellulose forms. If you imagine one stick of spaghetti, this is the equivalent of one long chain of beta glucose molecules creating that straight structure. If you gather about 60 to 76 sticks of spaghetti and hold them together, the free hydrogens in the beta glucose will form many hydrogen bonds between the sticks and create strength in its structure. The bundle of spaghetti represents the microfibrils. I hope that makes sense. You've got this diagram here that shows the ring form of beta glucose and where the glycosidic bonds form. Hopefully you can recall that the hydroxyl group is in the up position on the right hand side of the glucose molecule and in the down position on the left hand side as you look at the molecule. This means that you can get a diagonal type bonding between the two beta molecules, and actually this forces the second beta molecule to effectively swivel to accommodate this glycosidic bond. You can see in the bottom diagram that the bond is on the top between the first and the second molecules, and at the bottom for the second and third molecules. This is what ensures a straight chain is formed. Lastly, we need to look at glycogen. This is the main energy source material in animals, and it's mainly found in the liver and muscle cells. Glycogen, like starch, is a branched polysaccharide. It's a chemical similar to amylopectin in that it is composed of alpha glucose molecules, but there are more 1,6 bonds mixed in with 1,4 links. This means that glycogen has many more side branches, and interestingly, it is more water soluble than starch is. The high numbers of branches mean we can hydrolyze the bonds and release energy very quickly. This image shows you an image of glycogen in terms of its bonds and a representation of its several branches. So that's all I have for you on this topic area. My next video will be on tackling past exam questions and carefully studying the mark schemes to ensure you know what the key terms are to use in an exam. And then we'll be moving on to lipids. Hope you gained something valuable from this video. Please share, like and subscribe, and feel free to ask any questions in the comment section. Thank you so much for watching, and I'll see you soon.